So good morning all of you. Today we'll study about central dogma. I think that you must have heard about the term central dogma. It was first proposed by Francis Crick. You know Crick, Watson and Crick, who discovered your DNA structure. Okay, so let us start about the central dogma. Well, the concept was first put forward by Francis Crick. According to this concept of central dogma, DNA is the storehouse of all genetic information which are required in heredity. Okay. So, what this central dogma concept says? In this concept says that the flow of genetic information from DNA to proteins occurs through various biochemical pathways. What are those? Replication, transcription and translation. So, central dogma is the process by which instructions in DNA are converted into functional product. What are functional product? Ultimately the proteins. Okay. So, the coded informations that are found in DNA, they are decodified or decoded in the process of translation and ultimately proteins are formed. So, this is what the concept of central dogma is. So, as you can see in this picture, first DNA gives rise to another DNA through the process of replication, then DNA gives rise to RNA by the process of transcription. Okay. Now, the RNA synthesized contains, that is mRNA synthesized contains all the informations, coded information that were present in DNA. Now, these informations or these codes are being decoded in the process of translation to produce proteins. This is what your original concept of central dogma was. But later some new modifications were done to the original concept of central dogma. As in case of viruses, it was found that the genetic material is RNA instead of DNA, especially in tobacco mosaic virus. Not in all viruses, but in some RNA viruses like tobacco mosaic virus, RNA is the genetic material instead of DNA. Similarly, in addition to translation, transcription and DNA replication, additional information pathways have also been discovered. What are those? First one is RNA dependent RNA synthesis. We know that RNA synthesis is DNA dependent, but here in case of viruses, some RNA dependent RNA synthesis have been discovered. In addition to that, the process of reverse transcription has also been discovered, that is RNA dependent DNA synthesis. That means, for DNA synthesis RNA is required in viruses, in some viruses. So, due to the discoveries of these alternative pathways, and also the genetic material was not found in case of TMB as DNA, instead of it RNA was found. So, the central dogma has been modified a bit. That is how the information passes from DNA to protein. That is DNA replication. The original one was DNA replication occurs, then after that RNA is synthesized from DNA by the process of transcription. Then from RNA, through the process of your translation, proteins are synthesized. But when RNA is the genetic material, your RNA dependent RNA synthesis occurs, then translation occurs. Similarly, from RNA, DNA may also be synthesized through reverse transcription. So, this is what the central concept of central dogma is. So, the concept of central dogma says that the information pathways, that is how the information or coded information from DNA passes to the functional state, that is the product, functional product, that is the protein. So, this is what your central dogma is all about. Okay. So, I hope this video lecture will be beneficial for you. Thank you.